Buenos días, amable auditorio. El día de hoy, 17 de marzo del 2022, nos encontramos en el tercer día de la Semana del Cerebro que organiza la Facultad de Medicina y Psicología. Buenos días, amable auditorio. El día de hoy, 15 de marzo del 2022, nos encontramos en el tercer día de la Semana del Cerebro que organiza la Facultad de Medicina y Psicología de la Universidad Autónoma de Baja California. Soy el profesor Marco Antonio Hernández Lepe y seré moderador en esta sesión. Es un gusto para mí presentar a la doctora Morgane Frapin de la Universidad de Helsinki, Finlandia. A continuación, realizaré una breve semblanza de nuestro ponente. La doctora estudió en 2012 el primer año de licenciatura en la Universidad de Nantes, enfocada en biología, geociencias y química. El segundo año de licenciatura fue enfocado en biología y bioquímica. A partir del 2014 estudió el primer año de ingeniería en el área agroalimentaria, así también el segundo y tercer año, donde el tercer año también estudió un año de su maestría enfocado en nutrición humana. De 2017 al 2021 estudió el doctorado en biología de la salud en la Universidad de Bretagne, Loire. Ella también tiene una gran cantidad de artículos científicos publicados, además de algunos premios, ¿no? Siendo finalista, por ejemplo, en el concurso de la presentación de la tesis en 3 minutos que realizan en donde, pues, en esta área del mundo donde ella se encuentra. Y, pues, le pedimos al auditorio que si tienen preguntas, las hagan a través del chat de YouTube o en los comentarios de la publicación en Facebook, así como también los presentes les vamos a ceder el micrófono. Y al final de la sesión se van a responder todas estas preguntas. Le pido por favor a la doctora Morgane Frapin que inicie su presentación. Igual la doctora Sandra Olvera le va a dar unas palabras también.
Okay, thank you very much, Morgane, for to present this result that is part of the test the doctorate. The title of the conference is Fetal Malnutrition and Development of the Hypothalamus. So welcome to this event that the brain awakens brain. So can you start?
Yes. So up. So hi, good morning, everyone. So it's just a bit strange for me to say good morning because it's evening here in Helsinki, but good morning to you and thank you for the invitation. So I'm going to talk about fetal malnutrition and hypothalamic developments. The work I'm about to present was carried out during my PhD, so in France, under the direction of Dr. Valérie Amargé, in the physiological and pathology of nutritional adaptation research laboratory. And during my PhD, I tried to answer the following question: what is the impact of maternal protein restriction on the development of the hypothalamus in rats? So first, this question is based on the DOAD concept, so developmental origins of health and disease. This concept emerged in the 80s with the work of British epidemiologists David Barker and Clive Osmond, and it has been shown through epidemiological studies and starvation episodes that a non-optimal environment during the first 1,000 days of life, so from conception to the 2 years old, this non-optimal environment can lead to nefast effect on the health of offspring at short and long term. Among these non-optimal environments, we find fetal malnutrition. this fetal malnutrition corresponds to a non-optimal nutritional intake in the womb, so in the uterus of the mother, and with, for example, too much or not enough of essential nutrients. This malnutrition can be due to an unbalanced diet of the mother. So when the diet contains a lot of fat or a lot of sugar or not enough protein, but it can be also due to dysfunction of the placenta. So when there is a placental dysfunction, the nutrient have some problems to go from the mother to the fetus during pregnancy. So the fetus doesn't receive enough nutrients. This fetal malnutrition also have effect at short term and long term. So at short term, this fetal malnutrition increase the risk for the fetus, for the baby, to have a low birth weight. So this baby born with weight under 2.5 kilograms. And in the long term, there is also some consequence, like an increased risk to develop some disease like type 2 diabetes or obesity, but also some dysregulation of the control of food intake. So the person can't control what they eat, or have some problem to control what they eat. To study that, it's necessary to have animals models, because with epidemiological studies, so with studies done on human, when we check what happens to population which have a starvation episode during their life or stuff like that, we can see what happens after this, but it's difficult to really know why and how it happens. So there is many animals models to study that in different species. So in small species we can find rodents, so rats, mice, and rabbits. But there is also bigger animals model like sheep and pigs. In this model, the nutrient supply is mainly modified either by a change in the maternal diet or by a ligation of the uterine arteries. So when there is a ligation of this artery, there is less nutrients which goes from the mother to the fetus. In rodents, change in caloric intake, so more calorie or less calorie in maternal diet during gestation, or change in the specific nutrients, like more protein or less protein, or adjust the quantity of micronutrients. This changing is frequently found, is model frequently found. And the effect on the offspring is then studied at different life stages. So at fetus age, but also at adulthood and sometimes in newborns, and age between these different stages. So in my research, I used a model of maternal protein restriction during gestation. In this model, it is already been shown that there is an alteration in the birth weight of the pups, and as well as the development of insulin and leptin resistance, which are linked with obesity and type 2 diabetes sometimes, and an alteration in food intake later in life with hyperphagia in some case. Hyperphagia, so when there is an hyperphagia, they eat more than what they need. The pups eat more. So far I have explained why I was interested in the impact of maternal protein restriction during my PhD But now I will explain why I was interested in the development of the hypothalamus. So first, where the hypothalamus is located. The hypothalamus is a brain structure located on the ventral side of the brain, near so here in human it's here, so it's the brain and it's on the ventral side. It's the same in rats, it's in the ventral side, and this structure is near a part of the brain we call median eminence, so M here. The median eminence, so this part, is an area of the brain where hormones can go from the blood stream to the brain. So it's a key part of the brain because it's there is communication between the whole body and the brain thanks to this median eminence, and the hypothalamus is just above this median eminence. So as you can see here, so here is the hypothalamus, and you can see it's subdivided into several nuclei, which are connected thanks to nerve fibers, so neuron, axons, and communication. And on this hypothalamus, we can find specific neurons that can secreted hormones. So these hormones can after be released in the blood stream. Okay, so now the function of the hypothalamus. So the hypothalamus is involved in all the major physiological function of the body, like thermoregulation, so the regulation of the temperature of the body, the reproduction, the circadian rhythm, and the energy homeostasis. And these functions are associated with specific part of the hypothalamus. Some nuclei, which saw previously, are more involved in one function, and other are more involved in another function. So in the context of the DOAD, so in the context of my thesis, the hypothalamus is mainly studied for its role in the homeostasis of the energy balance, so the last one in blue. This function consists of maintaining the balance between energy intake, so food supply, what you eat, and energy expenditure, so your physical activity during the day. And it is associated with several hypothalamic nuclei. And an alteration of this balance, so when there is too much energy intake or too much less energy expenditure, this can lead to metabolic disorder, similar to those present during after a perinatal protein restriction, like the increasing of risk to develop obesity, type 2 diabetes. So the hypothalamus is able to detect information from all the body. I'm sorry if this figure seems really complicated, but I will try to explain it in an easy way. So the hypothalamus can detect stimuli from all the body, and after it integrates this stimuli, so what he received from all the body, and he can send back a response to the body to say you have to do more activity or you have to eat, you don't have to eat. The regulation of energy balance homeostasis, so the function of hypothalamus I'm interested in, depends of hormones, so that are in the bloodstream. And among these hormones, we can find ghrelin. It's an hormone secreted by the stomach. And this hormone leads to what we call an orexigenic response. So it means an increase of food intake. But it can also detect two other hormones, the insulin and the leptin, secreted by pancreas for insulin and adipose tissue for leptin. And conversely, this two hormone leads to an anorexigenic response. So this anorexic response is an inhibition of food intake, so you eat less. So this hormone cross the brain barrier, so they enter to the brain thanks to the ME, the part of the brain I presented you previously, and they can interact with two populations of neurons that are specific. One of this population give answer to increase the food intake, so the balance will go in favor of food intake, and the other population neurons decrease the food intake, so the balance go in favor of energy expenditure. So alteration in the structure of the hypothalamus have been observed following perinatal protein restriction. These consequences are heterogeneous between the different nuclei. And for example, the malnutrition leads to a modification of the neuronal density, so the number of neurons, but also the proportion of some specific neurons. So I presented you previously the orexigenic neurons, the neurons who can increase the food intake, and the anorexigenic neurons, the neuron which can decrease the food intake. So protein restriction can alter the proportion of this neuron. So, okay, and it also can alter some receptor specific to capture the hormone from the bloodstream. So if there is less of this receptor, this hormone can't be detected by the brain, so it can't have the response we want. And okay, so all of these changes are likely to be related to the alteration in food intake present in this model. I said previously that in this model we observe food intake modification like hyperphagia when people eat more, but we also observe change in meal structure. So change in meal structure is that people can eat the same quantity, but more often and like that. So it's not the same structure, meal structure. And to understand the alterations that can occur in the hypothalamus, it is important to understand how the hypothalamus develops. So how is the formation of the hypothalamus? So this formation starts at E10. So E10 is for embryonic day 10 in rodents, so in rats. So it's the 10th day of gestation. And in human, it starts the first week of gestation. The hypothalamus from neuron stem cell. So this kind of cell can is what we call undifferentiated cells. They can first proliferate to increase the number of cells in the brain. Then they can differentiate in several kind of cells. So as the other part of the brain, the hypothalamus, during the mid-gestation, this cell in the hypothalamus will differentiate in neurons. So the cell which gives the neurons in our brain, the cell responsible of communication in our brains. So first in mid-gestation, this cell gives neurons. It's called neurogenesis. And at late gestation, this cell will give glial cell, so astrocytes and oligodendrocytes. And this step is called gliogenesis. So astrocyte is these star-shaped cells, and these cells have many essential functions to neuron, like provision of nutrients. And oligodendrocytes are can create a spirally wrapped around the axon of neurons, and it increases the speed of the nerve's impulsion. Okay, so in rodent, neurogenesis, so the formation of neurons, takes place mainly during gestation. So here you can see it's a burst. We can also call burst P0 for postnatal day 0. So E is for embryonic day and P for postnatal. So neurons, neurogenesis, so formation of neurons, take place mainly during gestation, whereas gliogenesis, so the formation of astrocytes and oligodendrocytes, begin at the end of gestation and continues into the postnatal period. So during my thesis, I was more specifically interested in the early effect of maternal protein restriction. I carry out all my experiments on a rat fetus at the 17th day of gestation, so 4 days before birth. And this period corresponds to the transition between neurogenesis, so the formation of neuron, and astrocytogenesis, so the formation of astrocytes. So it's a key period, and it's an interesting period to know if everything goes well or not after a maternal protein restriction. So as I said to you, during my PhD, I used a maternal protein restriction model. And here I want to describe more in detail this model. So I use pregnant females, and these pregnant females were fed from the first day of gestation to the 70th day of gestation with a controlled diet containing 20% proteins, or a restricted protein diet containing 8% proteins. This both of these diets were isocaloric, so they contain the same level of calorie. That's why it's not a model where we study the difference in calorie between two groups. And the difference in calorie was compensated by an increase in the amount of cornstarch. So here you can see a rat fetus at 17 days of gestation. So it's really small. You can see there is a ruler just above, and it's around 1 or 2 centimeters. And we take the brain of this fetus, and on the ventral view of this brain, so here it's under the brain, it's the hypothalamus, so it's the part, my interesting part. So we perform some analysis at cellular level. So the cellular, we study the different type of cell present in hypothalamus, but also at molecular level, so inside the cell, the molecule present, so DNA, which is translate in which is translate in RNA, which is translate then in protein. So we study that in the hypothalamus directly after the sampling. So we take the hypothalamus and we study that. And after some day in culture, so after some day in vitro. So first, I investigated whether at cellular level the proliferation and the neurogenesis, so the formation of neuron, had been impacted by maternal protein restriction. So to do this, I measure the proportion of the different cell types present at this age. So the different cell type, it's like neural stem cells, so the cells that can proliferate, neurons, but also astrocytes. And then I use in vitro culture to measure the capacity of proliferation of the neural stem cells, so this cell, which has this specific ability to proliferate, but also to see if they can differentiate in neuron, astrocyte, and what they can do. So, sorry. So the hypothalamus cells collected for cellular analysis were mechanically dissociated into a cell suspension, and we count after. So we take the hypothalamus, we mechanically with the pipettes we create a suspension of cells which are really clearly separate in a liquid, and after we take a drop of this liquid and we count the number of cells we can see under a microscope, as you can see on this slide. So we count the cell and we compare how many cells we have in hypothalamus from fetus which received from mother which received a controlled diet and from mother which received a protein restricted diet. And what we found is that there is on average 15% less cells in the hypothalamus from restricted protein fetus than the one from controlled fetuses. And here, what you can see on this picture is in blue, it's the nucleus of the cell. So the cell, in the cell there is two parts, the nucleus and the cytosol. And in the nucleus we can find the DNA, and after we can find many stuff in the cytosol part of the cell. So the blue dot on this picture is the nucleus, so it's the DNA of the cell, and the red part is a protein called Ki67, and this protein is specific from neural stem cell. So like that, we know that the blue, if there is only blue, it's not proliferating cells, it's not a neural stem cell. But if there is red, it's proliferating cells. So we can count the number of blue, only blue, and the number of red cells. And like that, we can know how many proliferating cells we have. And with these techniques, we can't see difference between a protein-restricted fetus and controlled fetus. So at this stage, the number of neural stem cells is the same between our two groups. However, as I said to you previously, there is less cells. So it means maybe there is a problem before this stage. There is less cell at this stage, but the proportion of proliferating cell is the same. Then some of the cells have been fixed. So when I say fixed, is that it's like a picture. I put the cell and I just want the cell stay like this, and after I can do some more experiment on that. And another part of this cell was placed in a specific liquid, which contains specific molecule, which allows the neural stem cell, which allows this neural stem cell to proliferate, so to create more cells and to divide and create more cells. This liquid, this medium, is not made for neurons and astrocytes. So in this medium, these neurons and astrocytes die. But there is only neural stem cell which can proliferate and create what we call neurosphere. So we put the cell in this flask, and after 3 days in this incubator at 37 degrees, we obtain these neurospheres, which are a cluster of neural stem cells. So I also fixed, so I take this neurosphere, which I obtained after 3 days in the incubator, and I dissociate it in a cell again, and I also put it here to fix it. So again, to fix the cell, to be sure it's stay like that, and I can do more experiment on that. And so on this cell I fixed, so the cell obtained directly from the hypothalamus and the cell obtained after 3 days in the medium in vitro. I also did some I also tried to determine the proportion of the different cell types, so the neurons, stem cell, and mature and immature neurons, because we can also see the difference between mature and immature neurons. So here again is, as previously, in blue, it's DNA, so it's the nucleus of the cell. So each blue dot is a cell. And in red or orange is the specific marker. So in here, nest is for nesting, and it's a specific marker for neural stem cells, so the cell which have the abilities to proliferate. TOOP3 is a specific marker for immature neurons, and MAPE2 is a specific marker for neurons. So we can see at 17 days of gestation that there is this proportion, so 60% of cells which are the capacity to proliferate, and 34% and 30% of cells which are neurons. So yes, this immature neuron and neurons can express both. It's difficult to really distinguish. It's probably the same population, so we have about 30% of neurons. But we didn't see any difference between our two groups for this marker. So there is the same proportion of undifferentiated cells and same proportion of neurons between our two groups. So here it's at 70-day gestation. So if it's the hypothalamus, I take and dissociate directly. Here it's for the neurosphere, so it's for the cell I let 3 days in vitro. So you can see here for these cells, there is more undifferentiated cells, and it's logical because I select the neural stem cells, the cells which have the capabilities to proliferate. So there is more undifferentiated cells in this population than in the entire hypothalamus. And in this neurosphere, what we saw is that there is more immature neurons in protein-restricted group than in control group. So maternal protein restriction affect the capacities of the neural stem cell to differentiate. And at this stage, so at 17 days of gestation, these cells must give more astrocytes now than neurons. That's why there is probably a delay in neurogenesis when there is maternal protein restriction. So at this age, there is still the formation of a lot of neurons, but at this stage, normally we have to have more astrocytes. So I talked a lot about astrocytes, but I showed you only a picture with neuron and neural stem cell. It's because I tried to see the proportion of astrocyte in the hypothalamus at this age, so at 17 days of gestation. But at this age, there is no mature astrocyte. So here in this picture, you can see again in blue the nucleus, so the DNA, and in red it's neuron, so there is some neurons here, and in green it's astrocytes. And this picture was taken at birth. So at birth we can see there is a lot of astrocyte, but when I try to do this before birth, so at 70 days of gestation, I have no astrocyte. That's why I can't quantify and I can't say if there is a problem in astrocyte proportion at this age, because there is no astrocyte, and it's consistent with what I presented earlier on the development of the hypothalamus, where we can see that ED correspond to the beginning of astrocytogenesis, so the formation of astrocyte. So it's normal, there is no high number of mature astrocyte. So at molecular level, so inside the cell, I try to see the impact of maternal protein restriction on the expression of gene. So the DNA on the cells give the RNA, and this RNA give the protein after translation. So there is some RNA in the cell. So what I did is I extract this RNA from the cell and I sequence this RNA. So I just want to know which, so there is a lot of RNA, and I like this, it's little fragment of RNA, and I sequence it to know which fragment is that. And here you can see in this, for example, in this hypothalamus, we have 3 green, 2 blue, 2 red, and like thanks to sequencing, I can know that there is 2 RNA which came from the red genes and like that. So we detect 11,000 different genes in the hypothalamus. And what we did after, we try to see if these genes are more represented or less represented after protein restriction. For that, we compare the number of each gene, RNA, which came from a gene. So in this example, we can see that for control group, we have 3 of these green RNA, and we have 5 of these green RNA. So there is an overexpression. There is more of this in protein restricted group. For this one, there is less. So there is an underexpression. And for this one, it's the same. So that it's the same expression. And we found that there is 440 genes differently expressed between these 2 groups. So it's quite a lot. And on this figure, which can scare, it's just to show you that there is some of these genes which are underexpressed in restricted groups. So they are more expressed in the control group here on the left side. And on the right side, there is some genes. So all the red dots, all the dot is a gene. So all the red dot is the genes that are more expressed in the protein restricted group. So there is a lot of difference when we look inside the cell. What we have so is that among these genes, a lot of genes encode for protein of this mitochondrial respiratory chain. And this mitochondrial respiratory chain is involved in the production of energy in the cell. I don't know if you know the ATP, but it's thanks to this mitochondrial respiratory chain that there is this ATP, so it can give energy to the cell. And there is also a difference in the functionment of this this chain, respiratory chain. So it means that there is an increase in protein restricted groups. There is an increase of activity of this chain. And it's really interesting because it has been shown that mitochondrial metabolism play an essential role in neuronal differentiation. And this mitochondria, which are inside the cell, so they are in the cytosol, the shape of this mitochondria change during neurogenesis. So what we see can be linked with that. So we can't say more now about that, but it's good to know that there is a link between neurogenesis and the activity and the morphology of this mitochondria. What we also see is that there is a lot of gene involved in a specific molecular way. It's called epitranscriptomic, and I'm sorry, maybe it's a bit difficult now, but in the RNA is, in the RNA we have 4 bases, adenosine, tyrosine, guanine, and cytosine. And RNA is composed of these 4 bases. So we have A, T, G, C, T, G, C, and it's like that. And yeah, I made a mistake. It's not thymine in RNA, it's uracil, but it's quite the same that thymine we find it in DNA. And so the adenosine, so one of these bases, can have a specific part. Here I call this, it's right here in green, CH3. And we found that the gene which codes for the protein which can put this specific part on this base is differently expressed between our 2 groups. And this specific part, the CH3, can also be removed by some proteins which are also differently expressed between our 2 groups. So why there is this CH3 on this base? There is several functions for this, and it's to control the fate of the RNA inside the cell. So the RNA is created in the nucleus of the cells, and this specific modification of RNA can control the degradation of RNA if we want more or less of this RNA, so we can degrade if we want less. But it can also control the transport of this RNA in the cell. So if we want the RNA stay in the nucleus near the DNA, or if we want the RNA goes to the cytosol, or if we also want if the RNA goes to the axon from the neuron, so go really far from the nucleus. And for that, there is some specific protein which detect this modification and can say what to do with this RNA. And some of these proteins were also impacted by maternal protein restriction. So to conclude, here is a diagram of the main change I observed in the hypothalamus of protein-restricted fetus during gestation. So at the cellular level, the hypothalamus of the restricted fetus contains fewer cells and a greater proportion of cells destined to be neurons. And thanks to molecular analysis, so inside the cell, I also be able to see that maternal protein restriction possibly impact so the fate of RNA and also the activity of mitochondria, so the organelles necessary for energy production in our body. So this results shown an early alteration of the hypothalamus after a maternal protein restriction. However, it's still difficult to link this alteration at molecular level with what we can see in all bodies, so the increase of some disease and what we see in long term. So we still need to do some analysis at different age, different stage, and more molecular analysis to understand the link between what I saw during my PhD and what we can see at long term on this model. So I would like to thank my thesis laboratory for the help in carrying out all this research work, and thanks you for your attention.
Thanks for your conference, Dr. Morgane Frapin. Bueno, antes de continuar, le recordamos al auditorio que accedan al formulario que aparecerá en el chat en vivo para registrar su asistencia. Ahorita debe estar disponible en el chat. Ahora vamos a pasar al inicio de la sesión de preguntas. ¿Hay preguntas en el chat o no? ¿Aquí alguien tiene alguna pregunta? Bueno, le cedo la palabra a la doctora Sandra Olvera. No sé si quiere comentar algo.
So, thank you very much, Morgane, for this talk. I think that it's important to study, for example, I have a question. For example, I know that the nutrition during the camp induce or is a factor of risk for development of obesity, no, and diabetes. So in this case, your studies shows that maybe the difference is a nivel del hypothalamus, no? So there are some strategies for prevent this change that we can do in the maybe I don't know algún change the alimentation, also change the epigenetic maybe.
Yeah, if it's difficult to change epigenetic because if we change the expression of the proteins which create this epigenetic modification on DNA or on RNA, if it's epitranscriptomic modification, we for the moment we can't change it only on one part. So we really don't know what can happen if we change it. I don't know if I'm clear, but it's difficult to select one part and change it in only one part, and we really don't know what will happen if we change it a lot. It can have a lot of consequences that are not linked with nutrition or. I don't know if I answered to your question, but
yes, because for example, not here in Mexico there are big problem of the obesity. So, and now that the obesity have a vicious circle, no? So, but maybe I don't know if there are some students, but is using rats in with a model of obesity that show also epigenetic change in hypothalamus. I don't know.
I can't say like that, but I think there is some. I know there is some study on rats about obesity during sometimes maternal obesity. They study how maternal obesity can affect the descendants, and I think it can modify some gene expression. But like that, I can say really clearly what it changed. But yes, it's there is a link. And it's because epigenetic controls the gene regulation. It's really important in many processes.
Yeah, thank you so much. I know, ¿alguien se anima del auditorio por allá?
No, ya no hay más preguntas.
No, well.
La constancia, ¿no? Bueno, antes de finalizar, quisiera hacer entrega de la constancia a la doctora Morgane. Universidad Autónoma. Universidad Autónoma de Baja California, Facultad de Medicina y Psicología, otorga la presente constancia a doctora Morgane Frapin por su invaluable participación con la impartición de la conferencia Fetal Malnutrition and Development of the Hypothalamus, dentro de la Semana del Cerebro 2022, organizada por esta unidad académica. Atentamente, Tijuana, Baja California, 17 de marzo del 2022, por la realización plena del hombre, firma la directora de la facultad, doctora Julieta Yadira Islas Limón. Okay. Agradecemos su asistencia a este evento y les recordamos que siguen aún otros eventos más, tanto el día de hoy, con un taller de empatía, y también el día de mañana tenemos algunos eventos. Gracias.